The cell cycle of the eukaryotic cell is regulated by a molecular control system. This control system helps regulate the rate and time of cell division. Without this regulation, growth and development would be slowed, accelerated, or abnormal, resulting in complications. For example, skin cells are rapidly dividing our entire lives constantly replacing dead skin cells with new ones. Liver cells are capable of dividing, but only do so when a repair needs to be made. Nerve cells and muscle cells never divide in a fully matured body. A cell cycle control system directs the events of a cell cycle. This control system is a set of molecules that will trigger and organize important events in the cell cycle. It is similar to a washing machine because it works on its own with the help of a type of clock. Cell cycle will be regulated at checkpoints by internal and external signals. A checkpoint is a point in the cell cycle in which stop-and-go signals are given out to control the cycle. During the G1 checkpoint, sometimes called the restriction point, a cell will either get a go-ahead signal or it will not. If the cell is given the signal to continue on, it will finish the G1, S, G2, and M phases and will divide itself. However, if the signal is not given, the cell will leave the cycle and go into the G0 phase where it cannot divide. During the G2 checkpoint, mitosis may be set off. In order for a cell to pass the G2 phase, enough MPF molecules have to be produced. In order for this to occur, cyclin molecules that have accumulated will have to combine and work together with CDK molecules. After all this has occurred, mitosis may begin. MPF is a cyclin CDK complex. It is also known as the maturation promoting factor or the M-phase promoting factor. An internal signal may occur at the M-phase. Anaphase, which is when sister chromatids separate, cannot start until all chromosomes have attached to the spindle where the metaphase plate is located. Sister chromatids will stay together as long as some of their kinetochores do not attach themselves to the spindle microtubules. This will end up delaying anaphase. However, when the kinetochores attach themselves to the spindle, a protein will then be activated. The protein will then start a chain of events that occur at the molecular level. This will cause the cleavage of cohesins that will let sister chromatids part from each other. The whole mechanism is in place so that when daughter cells are created, they do not receive extra chromosomes or have missing chromosomes. An example of an external signal that gives a stop-and-go signs at each checkpoint. Cells do not divide when a certain nutrient is not found in the culture medium. Most mammalian cells will not divide unless the growth medium contains specific growth factors. Growth factors are proteins that cells release in order to encourage other cells to divide. A certain growth factor called PDGF, which stands for platelet-derived growth factor, is created by platelets or blood cell fragments. In order for fibroblasts to be divided in a culture, PDGF has to be used. Fibroblasts have PDGF receptors on their plasma membranes so that PDGF molecules can attach themselves to the receptors. This will begin a signal transduction pathway so that cells can go past the G1 checkpoint and divide. A cyclin is a protein that has a cyclically fluctuating concentration. A cyclin-dependent kinase or CDK, is a protein kinase that only becomes active when it has been attached to a specific cyclin. CDK activity fluctuates with the concentration of its cyclin partner. For example, MPF, or maturation promoting factor, is a cyclin CDK complex that peaks in activity as cyclin concentration peaks. MPF allows a cell to enter the M phase from the G2 checkpoint. MPF initiates the beginning of mitosis by releasing different proteins. MPF contributes to the cell cycle by aiding in condensing chromosomes and forming spindles in the prophase. Cancer cells escape the cell cycle control system and rapidly divide and invade surrounding tissues and good cells. These cancer cells do not adhere to anchorage dependence or density-dependent inhibition, instead creating their own required growth factor. Thus, cancer cells stop and continue to divide at random points and may never stop dividing if they have enough nutrients. The normal cell in a mammal would divide 20 to 50 times before they stop and die, but cancer cells are immortal and don't stop dividing. When cancer cells are growing far away from their origin area, it is called metastasis. Benign tumors are tumors that grow in their original area and are not serious and can be easily removed. Tumors that are invasive and grow away from their original area are called malignant tumors. These tumors are difficult to take care of and often result in a loss of organs and limbs. Scientists are learning more on cancer cells and how they can be controlled and treated by observing how normal cells function.